Hello friends, welcome to Targetneetjee. This is a revision series for NEET exam. Today we are here to discuss most important topic from chapter, molecular basis of inheritance, that is the lac operon. Before studying lac operon you must have to be familiar about the concept of central dogma. Central dogma was proposed by Francis Crick. This simply states that genetic information flows from DNA to RNA and from RNA to protein. In some viruses the flow of genetic information is in reverse direction, that is from RNA to DNA. This reverse process is termed as reverse transcription. For example, in retrovirus. Here is the diagram of central dogma which is given in your book. This diagram is most important from examination point of view. Formation of DNA from DNA is termed as replication. The formation of RNA from DNA is termed as transcription, and from RNA to protein is termed as translation. Now clear all the concepts of operon by watching further. By central dogma we may conclude that there is a unidirectional flow of genetic information. Now let's study what is operon. A polycistronic structural gene is regulated by a common promoter and regulatory gene. This type of arrangement is very common in bacteria and this is referred as operon. Now let's know how many types of operon is there. Operon is of different types. Some of them are lac operon, TRP operon, ARA operon, His operon, Val operon, and many more. Here we have to mainly focus on lac operon. TRP operon is tryptophan operon, which is a type of repressible operon. The operon concept or operon model was given by François Jacob and Jacques Monod. Here this is the diagram of lac operon when lactose, which is a inducer, is absent. During this process active repressor protein binds to the operator region and prevents the RNA polymerase from transcribing the structural gene or operon. Due to this prevention there is no transcription, no translation, no enzyme formation, and operon is in off condition. This all process occurs when lactose inducer is absent. The lac operon consists of three structural genes: the Z gene, the Y gene, and the A gene. Lac Z gene forms beta-galactosidase enzyme, which is responsible for breakdown of lactose into its monomeric units, glucose and galactose. Lac Y gene forms permease enzyme, which is responsible for increasing permeability of the cell for beta-galactosidase. Lac A gene forms transacetylase enzyme, which is responsible for transfer of acetyl group from acetyl coenzyme A to beta-galactosidase and increase its activity. And it also detoxify the byproducts which is produced during this process. Do you know? Here why lactose is termed as inducer. Lactose is the substrate for the enzyme beta-galactosidase and it regulates switching on and off the operon. Hence it is termed as inducer. Now see the culture medium concept. In the absence of preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cell through the action of permease. Then the lactose induces the operon. Point to be noted: a very low concentrations of expression of lac operon has to be present in the cell at the time, otherwise lactose cannot enters the cell. Now check if lactose, which is an inducer, is present, then what happens? In the presence of an inducer such as lactose or allolactose, the repressor protein is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Do you know? Glucose and galactose cannot act as inducer for lac operon. Lactose can act as inducer, but the true or real inducer for lac operon is allolactose. Now see the regulation of lac operon. The regulation of lac operon is termed as negative regulation. Lac operon is under the control of positive regulation as well. Give priority to negative regulation as compared to positive. Thank you for revising concept of lac operon with us. Click on subscribe button and press the bell icon for new updates.